Okay, so let's use that as the leverage point to talk a little bit about the pharmacology of the plant. Like it has a variety of different alkaloids, I understand, all of which seem to be related to a core alkaloid called mitragynine or something like that. What is the pharmacology of the plant in the sense of what chemicals are in it? What is its constituents? And then what do we know about how it's affecting our brain when we consume it?
Yeah, so that is a very interesting in regards to the core alkaloids that we know. We know that these are indole alkaloids. The indole core structure is preserved in some endogenous molecules that we have that we produce ourselves. So, for example, serotonin as a neurotransmitter has an indole structure. We have also a variety of other psychoactive substances that contain the indole structure. And I do not mean to at all put mitragynine and the other indole alkaloids that are present in kratom on the same level with some of these other indole ring containing alkaloids,
Sure, like
but
psilocybin, for example.
like psilocybin or LSD as a semi-synthetic, but they contain the same structure. However, they seem to have very different activities when it comes to which receptors they target in the body and what the resulting effects are. So when we talk about the two major alkaloids that have been studied, and we know at least structurally from their structure, we know at least 40 of them. So we currently mainly talk about two that are studied, but there might be much more to kratom that we simply do not investigate at the moment very well. But the two that are studied are mitragynine and 7-hydroxymitragynine. And mitragynine is primarily studied because it's the main alkaloid that is present in the leaf material. So mitragynine, what most people are focusing on is its activity at opioid receptors. And when I say activity at opioid receptors is, I mean, it interacts with opioid receptors, it binds to opioid receptors, and it acts as a what we call a partial agonist. And partial agonist means that it doesn't activate all of the opioid receptors it actually binds to. Um, so it activates some of them. It doesn't activate all of them. So the best comparison, if anybody would make a comparison, and it's kind of a little bit, it doesn't fit very well, but is buprenorphine, which is also it's a synthetic opioid that we use often in the treatment of opioid withdrawal to maintain people who are on opioid maintenance therapy, and uh, that is also a partial agonist at opioid receptors. The big difference that distinguishes mitragynine from the opioids like morphine or heroin or even oxycodone, hydrocodone, Percocet, and Vicodin, all of these brand names when I talk about just those, is what happens after they actually bind to the opioid receptors. So the opioid receptors, when we imagine a cell, let's think biology for a second, our biology high school classes. We have the cell with the cell wall, and the opioid receptors sit in the cell wall, basically. And on the outside, um, we have the binding site where our little mitragynine molecule binds to, or our morphine molecule binds to. And then on the inside of the cell wall, there's actually a little three-part kind of little trigger or a little switch. And that little switch is called a G protein. So that little G protein basically causes a little cascade of like a domino effect that happens after the receptor is activated. That switch turns on, and then this little domino cascade is activated upon the receptor being activated. But there's also a secondary cascade that actually happens that most people might not be aware of. And I wasn't aware of it until I learned more about mitragynine and these particular alkaloids. And that is the arrestin, beta arrestin 2 pathway. And the beta arrestin 2 pathway is responsible, is also another domino effect happens after that one is activated. And that actually leads to some of the not so beneficial effects that we often attribute to opioids. So the G protein coupled pathway that I mentioned, the domino effects from there down, leads to the pain relieving effects that we want to see, that we want to get out of it. But the beta arrestin pathway can lead to some of the not so beneficial effects, like the respiratory depression, for example. So slowed breathing that people experience that often leads to these overdose incidences where that can also lead to death in some cases, unfortunately. So what is the important distinction is when morphine or oxycodone, hydrocodone bind to the opioid receptors, then they activate both the G protein receptor cascade and the beta arrestin 2 cascade. When mitragynine binds, what has been confirmed in multiple studies by independent groups so far is that only the G protein coupled pathway is activated, not the beta arrestin 2 pathway. So that's an important distinction. Mm-hmm. We can, if you just basically look at does it bind to the beta opioid, does it bind to the opioid receptors, a molecule? You can study that in a, we now have very sophisticated programs that can do that. So you can study that on a computer. You don't ever have to actually go into an animal or even into a cell study. You can do that, and the computer will tell you, yes, it binds to the opioid receptor, but it doesn't actually, the computer cannot tell you what happens afterwards, after it binds. Does it activate both the G protein and the beta arrestin 2 pathway? We're not there yet that we can predict which particular downstream, we call that downstream effects, are mediated after binding of a particular molecule to a receptor.
So what I'm understanding here is that there's something unique about the alkaloids in kratom that bind to opioid receptors, providing analgesic effects and other effects, euphoria and all the rest, but that it doesn't suppress the breathing. So it sort of like provides some level of opioid activity without one of the primary dangers that's associated with acute use.
That is correct. Yes, that's basically what studies have found so far for mitragynine, for 7-hydroxymitragynine. We see that it is a little bit different. We are not certain what the exact mechanism is, but what has been shown in animal studies is that while mitragynine is not a reinforcer, so if you give morphine, if you give an animal a choice, if they want to self-administer morphine repeatedly, they will do so. So basically, they have a choice of a water bottle and a choice of morphine injection or saline injection and morphine injection. They will tap the lever that injects morphine. If you give them a choice of doing mitragynine, they will not repeatedly tap the mitragynine lever. They will not do that. With 7-hydroxymitragynine, they will do the same as they do with morphine. So there is a potential for tolerance and dependence development with 7-hydroxymitragynine, which we do not see with mitragynine. And we do not know exactly why that is, because it doesn't look like 7-hydroxymitragynine is also activating the beta-arrestin 2 domino pathway, that pathway that is activated once it binds to the opioid receptors. And that would also be a bad effect of many of the opioids, that they cause that tolerance and dependence development. So there's something about 7-hydroxymitragynine that we do not fully understand yet.
So the 7-hydroxy variety is able to doesn't activate that beta arrestin 2 pathway. Was that correct?
Correct. Yes. Yeah.
But that the activation of that pathway in other opioids is primarily what is associated to its dependency. But they both create dependency.
Yes. So 7-hydroxymitragynine also has been shown in animal studies to create tolerance and dependency development, but it doesn't seem to be mediated through the beta-arrestin 2 pathway. Yes.
Interesting. There's two questions. Okay, I'm going to start with this first question, and then I have another one specifically about what you're talking about. When it's activating the opioid receptors, now I understand that there's three different kinds of opioid receptors. One of them's kappa, one of them's mu, and the third one is delta. And primarily heroin, morphine, all the rest, it activates with mu, wherein something like salvinorin A from salvia seems to exclusively activate kappa opioid receptors. Do we have a sense of which opioid receptors are being activated specifically with kratom?
So I cannot talk for all of kratom. That's the problem, because we primarily look at mitragynine
right.
And 7-hydroxymitragynine. So for mitragynine, we know that it primarily targets the mu opioid receptors. And then it is I don't want to get this wrong, but it is also an agonist at the kappa opioid receptors, and I believe an antagonist at the delta opioid receptors. Whereas 7-hydroxymitragynine is also an agonist at the mu opioid receptors, but then an antagonist at the kappa and the delta opioid receptors.